So now we have Jason Ernst, who's going to be talking about functional genomic time series analysis. And Jason graduated from Carnegie Mellon with his PhD, and then went on to a postdoc at MIT. And in 2012, started here at UCLA with sort of joint appointment in biological chemistry, computer science, and bioinformatics. I like that, lots of titles. Excellent. Thank you.
Okay, so I'm going to talk about functional genomics time series analysis. So biological systems occur over time, and they've been studied quite frequently with time series gene expression data. And there's other types of data also emerging where experiments are conducted at multiple time points following some biological processes. And then the question is how to leverage this type of data. And traditionally, a lot of methods have taken time series data and applied the same computational methods one could apply if you just had independent static samples that could be like from completely different cell types, like you could apply some hierarchical clustering to the time series data. And the question is, could you potentially gain more insights or better use the data if you actually think about data that was actually collected over time and the data is probing some biological system that's dynamic. And this is giving you examples of a number of sort of widely used approaches. For example, the WGCNA method from UCLA. It's a very common representation of networks, and this is inferred from gene expression data and can often be applied to time series gene expression data, but it would ignore the temporal aspect of this data. So what I'm going to talk about in this talk is three methods, sort of give you an overview about them that one could apply to temporal data. The first two are some older work on, but it's still sort of widely applicable to modeling time series gene expression data. And then I'm going to talk about some newer work done by Pete, who's here, about applying a method designed to epigenomic data that's collected over time. So first, to talk about the short time series expression miner. So to sort of motivate this, when I was doing this work, there was gene expression data at the time was all microarray, but now this approach could also apply to RNA-seq. It was the case that about a third of all gene expression data that was curated into some of the major databases that collected microarray data were of gene expression, of time series gene expression data. And it was also the case that the vast majority of this data was relatively few time points. It was about 3 to 8 time points. And it sort of makes sense that you wouldn't have necessarily a large number of time points because these experiments can be expensive, or it can be hard to collect the samples, or you have limited access to the amount of material. For example, if you're taking blood from patients over time, you don't want to take it at too many time points, for example. So there's relatively few time points, and this sort of raises this issue that you're now profiling thousands of genes, and you have a limited number of time points, so you have a relatively small number of possible profiles you could have. So just by chance, you could have many genes that are potentially look like they're co-regulated, but it could just be what you would expect under just sort of random data. And what we need to do is sort of make more precise what we think of as a null model in this situation, and differentiate when is the co-occurrence of sets of genes having a similar expression pattern statistically significant, and when could it just be based on more random chance. And additionally, the clustering methods that people apply, like k-means and hierarchical clustering, they're not taking advantage of the temporal ordering of experiments. So one could permute the time order of the experiments you give it, and you get the same exact clustering. So that's a way to know the method is not using all the information about the experiment. And also, there's a sort of rich time series literature developed based on a lot of methods that assume you have much longer time points fitting various types of autoregressive or equations or other types of dynamic models. But some of these methods tend to break down when you have this few number of time points. And as what I was mentioning was, if you have thousands of genes and few time points, you're going to see many patterns just by random chance. And what I'm showing you here, these patterns might look like something interesting. Like you see lots of genes have this pattern, but this was actually something I generated based on applying k-means to simulated noise. So basically all values were just generated from an IID distribution in my data set, and I clustered them, and I get these types of clusters back on some data. So what would be nice is to have some way of knowing, is this seeing this type of pattern, this many genes having that pattern statistically significant in some sense? So this is a high-level strategy of the method we developed. So what we want to know is, are there significantly more genes having some pattern compared to the number of genes we would expect under a random ordering of time? So there's one true ordering of time where we have counts of how many genes have certain patterns, and then we can figure out an expectation under this model where we permute the order of time and then see how many genes have some profile. And one of the challenges with sort of this type of approach is that traditionally clustering, if you do a permutation of the time points and then you run a clustering, it's not clear how some clusters in one ordering would compare to your clustering under the original order. So we have a different strategy here, which is sort of a different twist on the clustering idea, is that first we define temporal profiles independent of the data. I'm going to talk more about each of these steps in detail. So the idea is, before we even look at the data, we're going to define a set of profiles that effectively cover the space of data that we would expect to see. Then we would assign genes to their most closely matching profile, these template patterns. Then we're going to compute also the expected number of genes we would assign to each profile. And then finally, we can determine which profiles we consider statistically significant. So now to talk first about this first step. So what we want to do is we want to define a set of distinct and representative model temporal profiles independent of the data. And one of the key things we're leveraging here is that we're making this assumption that we have a relatively small number of time points. So this is something which is sort of reasonable to do, that before we even look at the data, we could come up, for example, we have a parameter here of 50, we want 50 profiles, and this is a set that we're saying is covering this space. And we define it in terms of shape, so the units are arbitrary here. So how do we actually come up with a set where what we want is each profile we select to be relatively distinct from each other and to be representative of any profile that we would expect to see in the data? So what we do is, in this method, is we start with all shapes for which there's at most C units between any two time points of change. So between any two consecutive time points, you could either go up 2 units, go up 1 unit, hold steady, go down 1 unit, or go down 2 units. And then you have sort of that trajectory across each time point. And we exclude the straight flat profile. We make the assumption that we're going to filter genes which aren't differentially expressed at all between the time points. And everything is relative to the first time point. It's usually like a time point 0, and then you have some stimulus or something which you're monitoring relative to that. So we're looking at the expression change relative to that. So the y-axis would traditionally be like the log-fold expression change, and the x-axis are the different time points. And then what we do is we take, initially, we could take the most extreme, one of the most extreme profiles, for example, a profile which goes down at each time point. And then what we do is we select, we have a parameter k here, which is the number of profiles we want. And what we do at each step is or what our goal is we want to maximize the minimum distance between any two profiles. So we formally want to have the distance between any two profiles that we select, the closest between any two, to be as large as possible. And as a distance, we're going to use the square root of 1 minus the correlation coefficient. So then once we have one, what the method does is it greedily selects of our candidate profiles the profile which is most dissimilar to any profile that we currently have. And we do this iteratively. So that would be the third one selected, the fourth one selected, fifth one selected up here, and so forth. So then we would get these 50 profiles as our template profiles to be representative of any profile you would see here. And then this was optimizing specifically that any two profiles that we select are distinct from each other, but then one can also show that there's also theoretical guarantees that any profile we didn't select is also will be close to one that we did select as well. So now that we have a set of template profiles, what's nice about these is we define them independent of data. So the first thing we can do is, based on the real ordering of time, we can assign genes to their most likely profile. And then we're going to, at the next step, also do this based on random ordering of time. So these are our set of profiles. We assume we filter genes that aren't differentially expressed, and then we assign each gene to its most closely matching template profile. And what you see here are counts on the number of genes that were assigned to each profile. The number up here is just a profile ID. So for example, this is saying 150 genes were assigned to profile 43. Now the third step is, what we want to do is we want to get a calculation on the expected number of genes we see assigned to each profile. So what we do is if this was our real ordering of time, so you have a gene and then each column is a different time point, what we do is we construct permuted versions of the data where we permute the columns, and then we repeat the procedure, and we do this for either all the permutations which is feasible, or if it's long enough time series, we could do it for a subsample of the possible permutations. And then what this says is, on average, 28 genes get assigned to that specific profile. And again, the reason we're able to do this is we've leveraged the fact that the profiles were defined independent of the data. If we were just going in directly and clustering the expression, the clusters based on each permutation of the data would be different than the clusters from the original ordering, and it'd be hard to compare them. And now, because we have a count of the number that were actually observed and a count that are expected, we can compute statistical significance. And we assumed a binomial distribution, and we count sort of how statistically significant it is compared to the expectation based on the permutations of time. And now what I've done is I've ordered the profiles based on their statistical significance, and also one is like a post-processing step can color profiles that are both statistically significant and similar to each other, the same color. And then once you have a clustering or a set of profiles, you can go in and start comparing them to like gene annotation. So for example, it's saying that this profile was associated with negative regulation of cell death genes, and this was looking at a time series looking at an effect to pathogen stimulation. And then another one associated with DNA replication would go straight down initially. And just want to give you a quick demo of the actual software. So this is something to actually sort of broaden the use of some of the methodological approaches that can help having useful software. So what it's showing you here is the various profiles, and then they're colored if they're statistically significant, and we can order it by the actual significance. And then, for example, we can go into one of these profiles, see the shape, set of genes associated with it, and then also bring up what are the biological categories associated with them. And then also, if you're interested specifically in some category as well, you can go the other way too and see them ranked that way. Any questions about STEM? Yeah.
What would happen if you took those profiles that you created beforehand and did that same permutation sort of analysis, like pretending that they were also your set of genes? Would you be guaranteed to get sort of a uniform distribution for those profiles again?
So you're saying we take the candidate profiles and now assign them to the selected profiles?
Take the, not quite sure if I know your terminology, but the candidate profiles are the one that you selected in advance, right? Those guys.
So basically, so the candidate ones are sort of any of these, and then the selected ones, I was referring to the yellow.
Right, okay, so take your selected profiles and permute or jumble their time ordering, right? And then also assign them to the original unpermuted selected ones. Would you then get a uniform distribution? Or
So we have the requirement that everything starts at 0. So, we would be permuting, with the expression, we're permuting the original columns and then doing the log ratio. So you're
I see. So all your selected ones start at 0?
Yeah, it's a constraint. They start at 0, and then also we have this constraint that they have at most a 2-unit change, when if you permute the profiles, you wouldn't necessarily get this. And that sort of maximum unit change sort of enforces more of a smoothness that you wouldn't see dramatically different changes between 2 close time points. Okay. The next approach I want to talk about is DREM, the Dynamic Regulatory Events Miner. So STEM, it's good at identifying these clusters of patterns, but it's not giving you sort of a global view of the regulatory response. So for example, you might have something like this, where you have these 2 patterns, which are effectively returned as independent clusters, but what you might want to really think about them is they're really the same pattern up until some time point, and then you have a bifurcation event where some genes go higher than the other ones. And then another thing is you would want to start getting some more mechanistic understanding of what's driving these different patterns in terms of a dynamic regulatory network. You would want to know potentially like what transcription factors might be associated with some genes going higher at some instance of time than other genes. And specifically what we were interested in was integrating static data associating transcription factors regulating certain genes. So it can often be the case that you have dynamic information from the time series gene expression data, where you have each gene over each time point, but then in terms of another aspect of the regulation information for genes is what transcription factors, what proteins are associated with regulating them. And this type of information can come from a number of approaches. One is from ChIP-seq type data, where you have maps of where a specific transcription factor is binding, and if it's binding in proximity to a gene, you could say that transcription factor might be associated with a gene. Another approach is using regulatory DNA sequence motifs, so patterns in the DNA a transcription factor is predicted to bind, and then use that to predict it's associated with a gene. So the DNA sequence is inherently static, so you're not going to get dynamic information from that. And then from the ChIP-seq data, it's very hard to do time series of ChIP-seq data even for one factor, but then often you would want to consider multiple transcription factors, and usually you wouldn't get that type of information for or as many transcription factors as you would necessarily be interested in. So what we effectively want to do is elevate, in some sense, the static transcription factor gene association data to having a dynamic interpretation by integrating it with the gene expression data. So what you're seeing here is the gene expression data for heat shock response in yeast. So this is the individual expression data. And the method that I developed, DREM, is effectively building a map on top of this, where each line here corresponds to a different path through the expression space, a major path. And then you see the green nodes here correspond to bifurcation events. So it's effectively a tree structure here. And then it's saying that genes have sort of some pattern, and then there's some divergence. And then it's annotated these splits with transcription factors that are associated with potentially driving this split. So it's saying here that these transcription factors are more, genes that this transcription factor targets are more likely on this branch out of the node than this branch. And also, we can actually use the static transcription factor gene interaction information to learn a better structure and better model to begin with. It doesn't necessarily just need to be on the post-processing. We could find a sort of a more coherent view of the whole regulatory response based on all the information we have with the model I'm going to talk about. And this is showing you sort of what it looks like through these splits. So we have here in orange a set of genes associated to the lower split, and then now again through this split, and this split, and this split, we see these bifurcation events. And then here is this final split, and this is sort of a broader blown up view of that split, where we have all these genes which effectively have the same expression distribution until the final two time points. And then you see there's this bifurcation at the last two time points. These genes had this higher expression than these other two genes. And sort of the question is, what is this split associated with here? And it turns out that genes regulated by a certain transcription factor based on our static predictions of which transcription factors regulate which genes, which was generated by, actually it was ChIP data from Rick Young's group primarily. They're more likely to be on the higher path. So I'm showing you all the genes that are targeted by this transcription factor, and you can see that they much prefer this path up here. So that's suggesting that this transcription factor might be associated with this higher path, and we're able also to make this more specific prediction of the timing of when this event is happening, that it's having some influence at this time point, because that's where you see genes basically have the same distribution, and those genes that it's targeting go higher. And this is something that, even though we don't have any dynamic information about when this transcription factor binds, we can make a statement about that transcription factor regulating certain genes at a certain time point by integrating the static information with the dynamic time series information. And now, if you're familiar with the yeast transcription factors, this is a known regulator of the cell cycle in yeast. And it turns out that this has to do with sort of a synchronization of the cell cycle in response to stress and stress recovery. So we were able to make a testable biological prediction about sort of the state of the cell cycle in yeast in terms of a bud counting experiment. And what we saw was there was a spike in the number of percentage of cells that were in this no bud class occurring around sort of 60 minutes, which lined up quite well with this here. So we were able to, our collaborators made a experimental validation and some computational predictions that came out here. Now to talk a little more about what's the actual model behind getting this structure here. So first give you a toy example of this to illustrate this. So what we have here is this, where we have three paths. I mean, in practice, data won't be this clean, but this is just for illustration purposes. And what we have is sort of there's one bifurcation event here and one here. And what we're going to assume is the expression data is generated from Gaussian distributions based on some mean and variance associated with its sort of true expression along a path. So what it's saying here is that here these genes have this Gaussian distribution of expression, and the genes along this path have a Gaussian, and then along this they have a Gaussian. And then we also have transition probabilities. If you're in this sort of position here, then there's some probability you transition to going to this state up here, and some probability going to this state here. And then if you're here, some probability going to this state, and some probability going to this state. And if we, and we're going to assume that each of these paths have the same number of genes. So then if we didn't have any other information, the maximum likelihood probabilities we would assign is 2/3 probability of transitioning here, 1/3 here, and then 1/2 and 1/2. But now suppose we had additional information. Say we recognize that most genes regulated by this transcription factor A tended to be on this path. Then what we would want to do is if we saw a gene that we knew was regulated by transcription factor A, we would want to assign a higher probability that it would transition to this state versus this state, and then a higher probability to transition to this state, given that additional information that we know this gene is regulated by transcription factor A, and most genes regulated by transcription factor A are along this path. And similarly, if a gene was regulated by transcription factor B, and most genes regulated by transcription factor B are on this path, we would assign a higher probability of transitioning here. And then more generally, what we do is we have a classifier which converts the set of static transcription factor gene associations for a gene, so a vector of which transcription factors bind the gene, into a probability that it should go either to this higher or lower path. And we use a logistic classifier, so it gives those sort of well-defined probabilities. And what we would need to do is learn the weights. These are parameters associated with each of the input features. So what we're doing is we're taking this vector of, for a given gene, of which transcription factors bind it. So it'd be 0, 1 vector, we're going to assume for now, could be extended to associate activation and repression, has since been. And then what you do is you associate some probability of, so in this case, we're doing a binary split, but this actually generalizes and DREM supports 3-way or 4-way splits, and logistic regression also generalizes to multi-way classification. And what it does is it would give some probability of P1 of going this up path and 1 minus P1 going this down path. And this is a specific instance of something called an input-output hidden Markov model, where it extends a normal hidden Markov model in that the transitions now depend on additional input. That's not basically your observation. So you can think of it as you have some input here. So the input is like the static transcription factor genes, and that's going to affect the observations you see. And that we're allowing it to influence it through the transition probabilities. So just to go over some more details about how we actually go from this expression to this type of tree structure, what we want to do is we want to, first for a given tree structure, we want to find the best setting of the parameters that would be consistent with the data. And what we do is we're going to be summing over, so we can write a likelihood function, and what we're going to end up doing is we're going to be summing over all genes, and then we're going to be considering all possible paths through the model, and then a probability for a given gene and a given path, what's the product over all the Gaussian emission densities associated with that gene. So this would be sort of the emission probabilities of an HMM, and then the transition probabilities, which is what's the probability when we're in one hidden state transitioning to the other hidden state, and what differentiates this from the traditional hidden Markov models that were conditioning on the input. And I mean, DREM could still be run ignoring the input and just using the input as like an external validation. But if we use the input here, it'll influence these transition probabilities to be more consistent with the expression data. And these transition probabilities are based on a logistic regression classifiers. And now, this is all for one structure, but one of the challenges here is that we need to learn the actual structure, not just the parameters. So what I developed was an approach where we do a hill climbing search over tree structures. So what you do is you would start with a flat single chain and then search, considering adding splits at each node that doesn't have a split, or if you're allowing more than 2 splits out of a node, any split that hasn't reached the maximum number of possible paths out of the node, and considering adding a split, retraining the model, scoring, taking the best scoring one, and then continuing and until you sort of reach a where you can improve the scoring. And one issue here is that the data, as you add more and more paths, your likelihood can improve just because you have more parameters. So you also need to penalize the complexity. So there's a parameter which penalizes for how many nodes you have in the model, so that if you're continuing to increase likelihood, you also have to do so more than a parameter on the complexity of the model. And then at the very end, there's also a step which considers pruning away some of the splits and paths that might still be there just because of the local maximum it was in from adding. So now to give you some examples on other examples on real data. So before I showed you the heat shock response in yeast, we applied it to other ones, for example, amino acid starvation response in yeast. And what came out of this was one of the splits here was annotated with INO4, which was not previously known to be involved in this response. And the reason it was here on this map was it had been profiled under normal growth conditions, but they hadn't profiled where it bound specifically in this stress response because they didn't think it was necessarily involved. But sort of this, there's probably enough overlap of the binding sites between the normal and the amino acid starvation that it led to this hypothesis now that INO4 was active in regulator in amino acid starvation response, and making more specific predictions about what sets of genes and what time points it might be exerting influence. And our collaborators in Itamar Simon's lab did a new chip-chip experiment at the time. So this was the predecessor of ChIP-seq. It's similar, but it's based on microarray-posted sequencing technology. And they showed that there was greater binding in amino acid response compared to sort of the normal condition baseline. So it's saying in response to this stimulus, we saw more binding of this at the sort of relevant time point we were predicting. And we had since, in addition to applying this to yeast, I also applied it to fly development as part of the modern code consortium efforts, and in a collaboration with Zoltan Oldphase group, we applied it to new data the lab had generated looking at the aerobic anaerobic shift in E. coli. And there's applications of DREMs by other groups as well. And now to give a quick demo of this software as well. So this is actually I'll hide the nodes. So this is your original data, and DREM has learned this map on top of it, and then you can go click on some split you're interested in, and you can see the sets of genes associated with the split. And then one can, so if we hide the nodes there, and you can also right-click on one of these, and so it's just showing you some transcription factors that exceeded the significance threshold, but you can get the full list and more details about how many genes were going into a path and how many went on each path out of it. And you can also, you can adjust the options of the thresholding here, or actually, yeah. So you can adjust some of the thresholds here if you want to see more genes or fewer predictions, and also if you're interested in a certain transcription factor, like in this case GCN4, it'll display all the genes associated by GCN4 in the data. And there's other options as well. Other questions about DREM before I talk about the last part? Yeah.
So how long does the learning process take?
So that really depends on the number of time points, because it sort of essentially the space you're looking at scales exponentially with it. But then it's not quite as bad, because at some point, like, the regularization would be sort of a stronger thing and be hard to overcome that. But in terms of like smaller models, if you're working with like 4 or 5 time points, that could be done in half hour or something like that. If you're looking at something that's on the order of 10, 12 time points, then it could be a day or something. And then it heavily depends both on, one, the number of genes. Again, here's something you can get better modeling if you like filter genes that don't change much, so it focuses more of the modeling power on genes that are showing real changes. So the number of genes, the number of time points, how many splits you allow out of a node, whether you allow 2 or 3, and also how much you're regularizing the complexity of the model, because it'll stop with simpler models. And another feature of the DREM search is, while it's running, you can click at any, if you're not running it in batch mode, if you're running it in the interface mode, you can click at any time and see the current model that it currently has. So you don't have to like wait all the way to the end, and then you could potentially know that it's sort of good enough at that point. Okay, the last part is now thinking about a different type of temporal data. So if you were at my earlier talks, I've talked a lot about histone modification data and this epigenomic data. So these, just to remind you, we have chemical modifications to the histone proteins, and there's specificity in terms of which type of chemical could be a methylation, acetylation, which specific amino acid residues being modified, and how many copies. And there's this protocol ChIP-seq, which allows mapping genome-wide. And a lot of this had been done in sort of independent cell types. But now there's been sort of an increasing number of data sets where people are taking some mark and profiling at multiple different time points of some response or developmental process. So you might, for example, follow some histone modification at 5 different profiles. And what's unique here is we both have this temporal dimension, so what I'm showing you here, time, but we also have this spatial dimension as well. And what Pete had developed here is a method to sort of think about both the spatial and temporal information. And specifically, what we decided to focus on was sort of 3 major classes of spatial dynamics: expansions, contractions, and regions that hold steady. And the reason we were focusing on these 3 specific patterns is, one, they seemed to be sort of important classes of patterns that are interpretable, and then also with the limited number of time points and some of the noise associated with them, if we start allowing a lot of flexibility in what types of patterns we're trying to detect, it can be much easier to overfit some noise. So it encourages sort of more robust patterns that we're detecting. So the question is, how can we, in sort of a systematic way, go from this time series of ChIP-seq data into classifying and identifying these different types of patterns, whether places are steady, expansions, or contractions. So this is showing you at a high level how the approach works. So we start with ChIP-seq signal, and we're working now one mark at a time. And what we do is we convert this into binary peak calls. And this could be done with other programs like Max is what we use, sort of standard peak callers. And you get patterns like this. And then what we do is we group them into different blocks. So blocks here have peak calls that are effectively overlapping at multiple different time points. And then what we want to do is we're going to classify them into these patterns, and we're also going to potentially re-estimate what we're considering the true block boundary. So for example, this block we might say it's steady, but we didn't see the peaks perfectly align. But what we're going to say is this is just random fluctuation compared to here where there really is a change of the territory that peak is occupying over time. So what we're doing here in our modeling is we make this assumption that there's some true unobserved peak boundary that we don't observe directly. What we observe is some noisy observation of where that peak boundary is actually located. And then we also make another assumption that between any two time points, the true peak boundary could either hold steady. So if it was in the steady class, it would be at the same position, or it could move, and if it was an expansion on the right edge, it would move to the right. If it was an expansion on the left edge, it would move to the left, and then vice versa for contraction. And then we use a Poisson distribution to model how much it would move between time points. And the rate of that Poisson can vary between time points. And then we assume a Gaussian distribution for the noise of where we actually observe the peak boundary relative to where it was actually observed. And again, the parameter of the Gaussian variance can also be time point specific. And so that was just between any one time point. What I said was we're making this modeling assumption that things either expand, hold steady, or contract. And we don't require it to be from the beginning to the end. We model it from some start time point within the series to some end time point. So this leads to these various combinations of classes of the boundary dynamics when you take into account the left edge and right edge. And we enforce that we have the same prior probability of these symmetric classes of dynamics. So these are the various combinations we could see between the two classes. And now this is giving you sort of the general overview of a graphical model that represents our modeling here. So what we have is we have sort of a set of variables for the left side and a set of variables for the right side of the boundary. And what we have in green are the actual observations of where the peak boundaries are. And then the H variables represent the hidden unobserved locations of the boundaries. And then D represents the dynamics, so this could either be steady, expand, or contract. So the hidden state at some position at one time point will depend on the hidden state at the previous plus the specific dynamic. And then there's some distribution. This is based on a Poisson, how you go from that hidden state position and that dynamic to where we think the hidden state would be at the next time point. And similarly, this is the Gaussian going from the hidden to the observation. And then, as I mentioned, we have this assumption that across all the time points, we need to have the same class of dynamics. So we have a variable that enforces the trajectory. And then we have the left and right edge can have different trajectories. And then we have a prior on our probability of seeing that combination of trajectories. So we have this modeling, and then we learn all the parameters of the model based on the observations, and then for each block, what its most likely hidden position of each peak boundary is, and the overall dynamics of that block. So this is showing you the output of the method. What we have here is, this is the raw signal, and then in blue are peak calls for it. And then what the method does is, so it works off of the peak calls, and we get these now annotations of these different locations. So for example, it's calling this right here as a steady peak call. And there's some procedure that I didn't go into the details of how we can sometimes break up certain blocks into two blocks if they overlap split peaks. While it classifies this as a contraction, and then this is an expansion, this is steady, and we can also further have late steady regions or early steady, which aren't steady the whole time point, but while we do have peaks, they are steady. And just to talk about one result Pete's done other analyses based on this model. One thing which we were observing is if you look at transcription starts of genes, there's this asymmetry that sometimes we see this situation that over time we have more of the signal expanding into the gene body, or we have the contraction removing it. And now that we have this approach that sort of can systematically call boundaries as either being steady or expansion, we could analyze to what extent were we seeing expansions preferentially going in the direction of transcription over time, or contractions sort of getting shorter with across the gene body and then steady on the side not facing the direction of transcription. And what we found for several different histone modifications was a preference for expansions expanding into the gene body, as you're seeing here, and likewise for contractions. So just to acknowledge the various collaborators, so the last part I talked about has been Pete's work, and then the STEM and DREM were done with my PhD advisors at Bar Joseph, and then these were some of the experimental collaborators who produced some of the data or did experimental validations. And then a number of people continued to develop DREM and have published a number of extensions, which I didn't really have time to talk about. So any questions?